For this first portion of this lab, I'm mixing hydrogen peroxide and the liver blend with nothing else added. And as soon as the hydrogen peroxide touches the liver blend, the enzyme catalase in the liver blend causes the reaction between the hydrogen peroxide, which is the substrate, and the enzyme catalase. And basically, the two products of that chemical reaction are oxygen gas and water, and as I add it to the liver blend, you can see the oxygen gas being released in the form of bubbles. For this second part of the experiment, I'm now mixing the liver blend that I created using the blender and some vinegar together. And what I notice about this mixture is that it's very thin and runny, and it kind of makes it look lighter in color, and it's not very thick. It makes it very thin and runny and a little bit more pink than the mixture originally was. For this third part of the experiment, I'm mixing my liver blend that I mixed with the vinegar to make it more acidic in with hydrogen peroxide. So as you can see right here, I'm taking the vinegar liver blend and placing it on the plate. And then I will add the hydrogen peroxide to this liver blend, which actually produces less bubbles than the previous part of the lab did. And as I sat there and waited, the bubbles actually rose, but there were fewer than there were in the first part of this lab. For this fourth portion of the lab, I'm now mixing a teaspoon of the liver blend that I originally started out with, and I'm mixing that with a teaspoon of baking soda. And as I mixed the baking soda and the liver blend together, I noticed that it got thicker, but the color didn't really change necessarily, in my opinion. I think the consistency of the mixture just got thicker as I stirred in the baking soda into the liver blend. It just got thicker. For this fifth portion of this liver lab, I'm now mixing the baking soda, liver blend, and hydrogen peroxide together. And I actually get a very unexpected reaction. I thought the reaction would be less, but immediately, as soon as I added the peroxide, there were a bunch of bubbles that formed, and they formed really quickly. I didn't have to wait at all. It was almost instant. For this sixth part of the lab, I'm now going to heat a teaspoon of the liver blend. For this part of the experiment, I'm now going to add hydrogen peroxide to my heated liver mixture. And what I noticed as soon as I heated my liver is that it actually turned green and started smelling really bad. When I first opened the liver container, I actually didn't really notice a smell. But as I added the hydrogen peroxide to this greenish, really dried out liver mixture, bubbles formed immediately, and a lot of them. And actually, I saw a lot of water coming out of it, meaning that the reaction occurred quickly with the catalase and the hydrogen peroxide, because I could see both the water and the oxygen bubbles form really quickly, faster than all of the experiments I had done before. In conclusion, what was actually kind of surprising after I had already done that one assignment we had had where we watched the Beverly biology guy do this experiment, and the heated liver didn't seem to have a reaction at all, but I noticed that the heated liver had one of the biggest reactions. So I went back ahead and redid everything just to see, and with one drop of each, being really precise and careful. And I actually found that that was just a weird outlier chance on the last one, and that the heated liver doesn't actually produce that big of a result, but for some reason it did when I tested it. So in my conclusion, as you can see right here, the normal one with nothing added to it had the biggest reaction, and I noticed that the acidic one had the most amount of small bubbles, meaning the bubbles were really close together, so it was kind of a solid white color. The base didn't have much of a reaction, but the bubbles that it had were close together, and the heated one didn't have much of a reaction at all.